The overall goal of this procedure is to characterize the influence of embryoid body size on RPE differentiation from iPS cells for directing embryoid body differentiation towards the RPE lineage. This is accomplished by first using the microwell technique to generate homogeneous embryoid bodies of defined sizes. In the second step, directed differentiation of the iPS cells is initiated towards the retinal lineage, and then the iPS RPE cells are identified and isolated. In the final step, the iPS RPE cells are enriched and expanded. Ultimately, the iPS RPE cells can be analyzed for their expression of iPS RPE specific markers.
The main advantage of this technique over existing methods like spontaneous differentiation is that our technique results in higher yield of RPE. This method can help to optimize the directed differentiation of iPS cells towards the RPE lineage.
The implications of this technique extend toward the therapy of retinal degenerative diseases such as macular degeneration, retinitis pigmentosa, or Stargardt's disease, since the replacement of diseased RPE with healthy RPE derived from iPS cells may be the best option for preventing loss of vision.
Generally, individuals new to this method will struggle, because stem cells are very delicate and subtle changes in their handling can affect the differentiation process.
Visual demonstration of this method is critical, as the embryo body harvesting step from the microwell plate is difficult.
Prior to seeding, warm the feeder-free stem cell culture medium to